The lack of scientific evidence for abiogenesis, or the abrupt life from non-life, is one of the strongest elements that Christians have as an argument for creation and God. Watch this conversation and comment and let me know what you think. God bless.
The Miller experiment was a tremendous advance because it was the first time somebody stopped talking and actually went in the lab and tried to do something. It created a lot of excitement, but it's been 75 years and we haven't gone very far.
I say this all the time, man, I am so glad you said it before I did. And this is a guy who worked with Stanley Miller. We may be able to approach the target a little bit more, but what happens is the target moves much further away from us because we learn the complexity of what is in a cell.
The amount of discoveries in biochemistry that have happened since the Miller-Urey experiment changes the game. It's so much more complex than the target they had back in mind back then. The structure of DNA was announced
right about
the
that
same
time.
year.
Yeah, right about that time.
As the Stanley Miller, 1953. The target that Stanley had when he started his experiment, they didn't even know what DNA was. And that's how juvenile the field of biochemistry was. It's grown up a lot. And with each new discovery, the target for abiogenesis gets further away. If you look up the Miller-Urey experiment, they'll always show you the equipment before it started, which is this gleaming glass apparatus. When the reaction is over, the inside of the glass is covered with this red, oily compound. And around the electrodes is a black carbon deposit. And that's where most of the carbon winds up. The amino acids they find are very dilute in the solution. And so Miller, actually did the experiment over time and took samples as he went. And he noticed in the beginning, the amino acids would build up in concentration, and then they would plateau for a while, and they would go back down at the end as the synthesis reaction slowed down. And so obviously something else was going on. It is, as you say, the amino acids are reacting with sugars that would be produced.
Look how easy it is to make great globs of this stuff, the molecules of life.
Most people think you get these amino acids and that's all that you had there. And then if you needed it to be chiral, they would selectively crystallize out. And then those crystals would find themselves all purified and ready to go. These are such sludges that they're totally unusable.
You basically get what we call a tar in prebiotic chemistry, which is just an undifferentiated mess of a whole bunch of organic molecules that we can't identify.
We can't even use them in our laboratory, let alone on an early Earth being done in a pool somewhere on the side of the ocean.